Alrighty, let's get going, folks. So now we are covering the evolutionary context of the immune system. This is kind of a weird lecture. I was just thinking when I was going through all the microbiome stuff that it's just cool to see that relationship, and I wanted to look into how that evolved over time. So immunity did not evolve to be antagonistic towards microbes, right? That wouldn't really make sense if you think about like early life, the primordial soup. Uh, Cells forming in that nonsense. Like everything was microscopic in nature. So microbes being enemy incarnate wasn't really the deal because evolution began in this microbe dominated world. So as we're going into understanding the different branches of immunity, I already kind of had you guys pause and think about the self versus non-self and the danger model and the idea that both of those are contextual and graded and that we have to consider both of those facts, the foreignness and the damage potential when assessing threats. So it's not merely fighting relationships, but it's not merely fighting infections, but managing relationships. I think that's a better way to understand what the immune system is accomplishing here, especially when you consider our commensal microbe friends. So balancing protection, tolerance, and cooperation. So like thinking about a time pre-immunity when everything was, that didn't have that level of multicellularity to it. So amps and toxins were the earliest form of defense. I want you to think like penicillin is just something secreted by bacteria to prevent other bacteria from getting in its territory. That kind of stuff. All right, so not the most complex sort of defense, but a toxin nonetheless, and more so just get away from me, so I can do my own thing, so I can get my own resources without threat of starvation. And then you guys might have heard of CRISPR-Cas9. We're not going to go super in depth by any means. If you're interested, look it up. It's basically a bacteria's means of genetic editing. So, like, I don't know, if you think of a bacteria's genome as just a loop of, well, that's a little too thick, like a loop, and they keep coming into contact with like some repeat infections, or they keep having trouble with this one type of microbe, they can edit their genome, right, and put something in there that might provide better defense against pathogen X. So don't think about this too much. I'm just trying to give you the early idea of where immunity came from. The earliest was amps and toxins, which are still used to this day. And we don't necessarily use gene editing, but we do use the sheer number of genetic combinations we can make to leverage our immune system. And that will make sense as we go along. But this is where all of this kind of started. So ancient innate immunity obviously emerges with multicellularity. We need that complex structure, that complex organism, because if we have cells, just dedicated to fighting infection, well, that must mean all of the other portions of life are covered. So you need to be multicellular to have real like immunity in the way we think of it. And it emerged in sponges, worms, and humans. And obviously those are very different branches of life, which is really cool. So mechanisms like, of course, phagocytosis, PRRs, which is cool. Those are the receptors, the ears we were talking about, and inflammation responses. We see pretty conserved reactions across wildly different branches of Mother Nature. Like a great example is sea urchins have a wide variety of PRRs. So those receptors that detect the ow signals, I'm sure that there is some genetic differences there, obviously. But the fact that they have stuff that is in the same immune family as us, I think is pretty cool. We're all connected. We're all connected on this planet. That's an Earth. Okay. Then how do we get to adaptive immunity? Because if it hasn't set in for you guys yet, I keep using the same example, but I think it is a good one. So we have our B cell with the antibody on it. And whenever we have that B cell meet the love of its life, which is its one and only bacterium or antigen or whatever disease that it's fighting, it only has one. And we keep saying Jim. So I guess I'll just keep going with it. I always write Jum. There we go. Oh, goodness. There we go. I can't express to you how crazy that is that there's that one-to-one relationship. You have a B cell that's ready and willing to fight Jim. You have a different B cell that's ready and willing to fight strep. And again, a different B cell that's just waiting for chickenpox to come in and try you. And I could keep going on and on and on. There's that one-to-one relationship. So that's why adaptive immunity takes a bit of time to get kickstarted. But how does that come up? Well, we actually found it appearing in gnathosomes, which is jawed vertebrae. So these weird, prehistoric fish. That's how far back this concept goes, which is so cool to me. But what do I mean by that? How do we even know that? So specificity relies on gene rearrangement. Gene rearrangement. That's the only way I get a B cell with a receptor that this one attacks Jim. But this receptor is going to be a little bit different. It's still going to be an antibody, but that one attacks strep. And this one, it's still an antibody, but it's going to be a little different, and that attacks chickenpox. So how do I get that level of specificity, of randomness, that I have a B cell that can fight anything that I come into contact with? Again, genetic rearrangement. And we're not going to get into the weeds too much. We'll talk about it more in Unit 2 on whenever we're growing B cells and how those receptors get made. Again, I don't want to go to biochemistry in this class, but what I do want to stress is one of the ways that we mediate gene rearrangement is through something called the RAG enzyme. Full disclosure, that's more so with T cells than it is with B cells, but it is a really cool concept that we found a process of gene rearrangement that we literally use, that our cells in our own bodies today use, all the way back in fish from 500 million years ago. So there you go. The gene was coded all the way back to that evolutionary split. Pretty cool. All right, so adaptivity provides specificity, like we said, one-to-one. Memory, which we haven't talked too much about. I'll talk about that in just a sec. And flexibility. So that means, whatever we come into contact with, odds are I'm going to have a cell tailor-made to fight it. Another cool thing with adaptivity and the specific response, I have definitely referenced that you make, once ever the B cell that's fighting the pathogen that's infected you gets activated, so now it's activated. Yeah. Okay. There was two things that happened. I want you to think, what are those two things? If I'm infected with something that only this guy can fight, well, I'm going to want a lot of him. So the first thing is clonal expansion. I make a bunch of that. And the other thing was they just start secreting a bunch of antibodies. A bunch. I'm talking like thousands per second. It's a ridiculous amount. What's cool about adaptive cells is a lot of them do this antibody, factory pathway, but some of them hang back. Some of them hang back and become like a B cell with glasses. Those are the studious ones. They hang back. They're not going to go fight just yet. Their goal, they are memory. Memory B cells, and you also have memory T cells too. This is the same thing for those as well. They are hoping and praying that whatever infected you tries to come back and do it again. because now we don't have to go through the whole dendritic cell, bridging the innate to adaptive, none of that stuff. Now I have a population of cells that know whatever this was fighting. So I already made a bunch of them. A good chunk of them stay back and hope that it happens again. Because if it does happen again, now I kind of have the pros of both responses. So remember, innate was fast, nonspecific. Adaptive, slow, specific. But now with memory, secondary infection, fast, and specific. So I kind of like that. Mother Nature has built a pretty cool immune system that says, I dare you to try again. I dare you to try again, because I left some scholars behind that can't wait for you to come back and try. So that's why adaptivity is so cool, and that's why we spend so much time talking about it. Is because that's what gives us a lot of our longevity, those memory cells. We got two more? Yeah, two more. Okay. So back to kind of the vibe I've been talking about. We need cooperation, right? Complete sterility is not helpful. These bacteria can provide a lot of stuff that we've already talked about. Nutritional supplementation, so that's like vitamins. Barrier fortification, so making sure my mucosa walls are goodness, like cemented down and strong and don't let anything in that I don't want in. And competitive exclusion. So even if they're not even providing me vitamins or nutrients or whatever, they are taking up space that a toxin or toxic microbe might. So again, it's the landlord thing. I would love a tenant that brings me cookies. That's fine. I would also love a tenant that I never hear from. That's fine with me too. So organisms that elected to tolerate microbes were selected for by evolution. That just shows with what we have now. We have all the successful organisms have some sort of relationship with their own microbiota. So whenever I say microbiome, I've been referencing that a lot. It's just the totality of microorganisms and the collective genomes living on that host. All right, so with us, I mean, it's in the order of trillions, if not quadrillions. There's so many microbes helping us out on our skin and in our gut. And they kind of represent a functional organ system. So it's dynamic, it's metabolically active, and they're integrated into our anatomy. So you can make an argument that it's kind of its own organ, which is kind of crazy. I'm not worried about that, though. That's more semantics. So let's talk about the mechanisms, though. And we've talked about these a good bit, but let's dive in. So competitive exclusion, this is the whole quiet tenant. They occupy ecological niches. So same thing, like in the woods, a bear is a predator. In the ocean, the orca whale is going to be that apex predator. We don't need another apex predator. That niche is taken. I'm oversimplifying, but that's kind of what we're after. If we have a bacteria in our gut that's already taking a resource or already providing something for that ecosystem, then we don't need anything else to do that. And if the bacteria fulfilling that ecological niche is commensal, it's nice, then we're perfect. So that's one of the benefits there. So yeah, preventing that physical attachment and making sure the nutrients are going to something that is benefiting us. So those are going to produce some amps, ideally that deter other microbes that might do us harm from joining. A quick note, barrier fortification. There are some microbial compounds that enhance tight junction integrity. That's junction. I'm sorry for my chicken scratch. So yeah, that just means my mucosal, the epithelium that makes up the walls of my gut tube, they're just going to be stronger now. Immune modulation. Again, we've said that a bunch. Selecting for Treg cells. That's awesome. We love our immune system and its power, but sometimes it is too powerful, especially when we have commensals in the area that do have, LPS and other PAMPs that are associated with pathogens. It's nice to tell the immune system to calm down sometimes. And I already said this as well. Vitamin synthesis, sometimes some bile acid metabolism, but just very helpful in breaking things down. The whole definition of vitamin is a vital amine that we can't get, that we don't produce in our natural biology, but it's vital for our longevity. So having an organism within that helps us do that is really cool. So yeah, we love our microbiome. All right, last thing, and this is probably the most important part of this lecture. Dysbiosis, the disruption of microbiome due to lifestyle or disease. My goodness gracious. So this can be due to a variety of things. You can have, your antibiotics sometimes can wipe out your commensals, especially in like more older populations that don't have as robust of a system to keep those commensals happy. Low fiber diets can certainly add to that, as many of your bacteria feed off of fiber. And a really cool hypothesis, and I referenced this in class when I was talking about the eosinophils, is the hygiene hypothesis. Our clean and modernized world might prevent introduction to some of these commensals. Like, yes, they're bacteria, but now that we have a really solid understanding of germ theory as a species, we're pretty sterile, and we have clean surfaces, and we don't want. Whenever we're having kids around, we don't want to expose them to anything bad. We don't want a sick child. That's bad, especially if it's your child. If that's the last thing you would want. But maybe there was some benefit. Maybe we had some old friends, microbes of yore that are getting wiped out by albeit our beneficial standards of hygiene. But maybe some of those microbes might have helped prevent allergic and autoimmune issues that are so prevalent today. That's just a theory, but it's fun to think about that we did evolve alongside all these microbes for X amount of years. So having the split that we're having from them may be the explanation for some of these negative diseases that we're seeing. So the disease is associated with dysbiosis, with a messed up microbiome, irritable bowel syndrome, IBS, again, some allergies and some neuroinflammation. I say the most common would be IBS. That is just the idea of your gut flora is thrown off, and you're getting poor digestion. And that can lead to a bunch of things. Obviously, diarrhea and things of that nature, poor nutrient uptake, all that kind of stuff. So, yeah, it's very important that this is held to a good standard and that we treat our microbiome well, because it does provide us many benefits and in many ways does prevent a lot of diseases of the gut. So, yes, please do remember dysbiosis and why that's important. All right, that's it for this lecture. That was pretty quick. Next up is going to be the breakdown of the various diseases as they pertain to the different types of barriers. And that'll make sense on that next lecture. But thank you very much. And I'll see you guys next time. Adios.